Okay, good morning, everybody. Happy Friday. Any questions before I get going with today's stuff? I am going to start with, and I don't know what happened on Wednesday. Apology. We covered all sorts of stuff, and then I went to post the lecture. And apparently, in all of the chaos of my little owl not communicating or not being plugged in, I don't think I ever recorded it. So there was no lecture to post. For those of you that will be tuning in to see these recorded, and you see this one, technically chapters, or not a few chapters, a few slides. You're like, a few chapters, what happened? A few slides ahead of where we left off on the recording from Monday. So go through the PowerPoints. If you got questions or concerns, for those of you that are just watching recordings, please shoot me an email and I'll get you up to speed if you've got any concerns about that stuff. So with that said, we left off thinking a little bit about some details of different organisms, and we're showing some staining as we move through here. And I think really this is kind of a good point for us to be beginning today's lecture, because really next week in the lab, this is what we're going to start thinking about, is some of the staining of different organisms. And one of them that I think is awesome to work with, especially when it's like the first go around with trying to stain stuff, are some of these guys, some of the bacillus. So we'll talk a little bit about how it looks on there in just one second. But bacillus, and there's we've already talked some bacillus as we've moved through here. There's numerous species, and Bacillus cereus is an organism. It's something that can be found out in the environment. The spores are potentially on all sorts of vegetables and crops and just things, all right, out in nature that potentially we could come in contact with. We could definitely isolate these things from just some of, like you did environmental swabbing. You potentially could find some bacillus because it's really all over the place, right? And dependent upon circumstances, some of these things do set the stage for causing some illness, but usually we just think of them as environmental organisms and not really that big of a deal. Cereus does have some potentials to cause foodborne issues, right? So we're looking at a gram stain of Bacillus cereus, a gram-positive rod, and Escherichia or E. coli. Escherichia coli is a gram-negative. And I like this, where it's got both of them on there. I will tell you, in the lab, we're going to, hopefully, we're going to give you cultures that are just one thing. And I think it's a little easier when you first start staining stuff up to know, I'm looking for this one thing. It should look like this, right? Rather than just throwing you a tube and saying, oh, stain it up and see what you see. Then you don't really know, right? It's kind of better to walk in there with hopefully knowing this is what I'm trying to find, right? Making it a little bit easier for you, maybe, all right? But I think that's good. So the bacillus, these guys are really, compared to E. coli, they're monsters. They're a lot larger. And gram staining them, if it's one of the first things you've ever stained up, pretty easy to find. And really, you know, they stick out a lot better than the faint E. coli as it often appears. It's so small and it's, you know, barely kind of pinkish colored. And it's, as far as trying to see shape with that, it's pretty hard to differentiate sometimes, right? With the bacillus, usually it's considerably easier to see nice long rods, right? I want to say something about that as well, as we are looking at some of this, right? the bacteria are very small, right? A couple of microns long, potentially, right? Really not that long to begin with, all right? And as we think about making a slide, you make your smear, you air dry it, you heat fix it, you do all of those things. So when we add the staining reagents, the stuff stays on there, all right? If that bacterium is spherical, if it's a coccus, well, it's going to be a bunch of little round balls that are all over on there, right? That morphology is easy to see, all right? But if they are rod-shaped or bacillus-shaped, the majority of these guys, they're going to be laying down flat on the slide, like the bacillus that we see here. Pretty easy to visualize, all right? The E. coli that's quite a bit smaller, well, most of those guys are laying down flat, horizontal on the slide, but a few of them are standing up on their ends. And sometimes we look at these things and see little speckles, little pink dots. And here it's really kind of blurred out. We're looking more at, well, really it's more the dark bacillus we're seeing. But some of these little speckles in here are those E. coli just kind of standing up on their tips, all right? If you've got just a culture of E. coli, you're going to see a lot of pink all over the place if you got a good smear, right? And you'll see some rods laying down horizontally, but you'll also see things that look smaller and they look like cocci, all right? But it's most likely just the end of E. coli that you're seeing and on the side, all right? So that definitely can lend to some confusion. And E. coli is so little that it's one that really kind of does that. The bacillus, typically they just kind of lay down, all right? And it's something that I've looked at these things and I'm like, I wonder really what's happening. And I'm like, it's got to be something like that. That makes sense that these guys would be doing something like that. So shifting from the gram stain here, this is a mixed culture. So with the gram stain, you could see that there's two things in there, two players, right? Maybe even more, really. I mean, you couldn't look at this and just say with confidence, this is only one type of bacillus or one type of gram-negative rod, right? The morphologies look pretty similar, right? As far as the positive rods and the negative rods. So you might be pretty confident that it's just two, right? We could try to grow these on a petri dish, grow them on a plate. We could do some streaking for isolation, which is what we've already kind of started thinking about with these plates, right? And if you had a mixed culture like this and you did good streaking for isolation, you would hope to find some colonies of bacillus and some colonies of E. coli, nice and separate and distinct. If they're right up on one another, even though they look like they're separate, if they're really close, it's hard to say that they're actually isolated and distinct. Many times when we get a culture that's mixed and we streak it for isolation, we're going to streak it for isolation one more time. We'll try to pick off two of these distinct colonies and replate in the hopes that we get a plate that truly is one thing or the other and not a mixture of the two, all right? So here we're really only looking at the bacillus, all right? In this case, it's growing on a blood agar plate. You can see the nice red with this plate. There's actually a little bit of discoloration there because the bacillus can be, some of them can be a little bit beta hemolytic. Bacillus cereus does have some hemolysis capabilities. And you can see some discoloration around the edges of this. It might be a little more partial breakdown. We call that alpha hemolysis. So the bacillus, and they're not all the same as far as how they are growing on these petri dishes, but they do like to really kind of spread out and take over, right? And we can see these big colonies that really just sort of blow up. And if you're trying to streak for isolation, if you're trying to get both E. coli and bacillus on there, and you let this plate grow for a couple of days, the potential for the bacillus to completely overwhelm that plate and grow on top of where E. coli was is definitely there. We call the bacillus a swarmer. So they've got flagella, they're able to move through. E. coli's got flagella too. But the bacillus does a really good job of kind of taking over and replicating really rapidly, especially if you give it a diet of blood. right? It will definitely thrive in that type of a habitat, right? So a couple of differences. And again, it would be, I guess, kind of be nice if we could see a plate with the two trying to be separated out. You'll see some of that in the lab. So here, we've got some spherical organisms. The Staphylococcus aureus does have kind of these clustered appearances. They say grape-like, all right? This is an electron micrograph, so a scanning showing us the surface of these cells. And I really I like this one, but we see a lot of electron micrographs. Lots of times the colors that we're seeing are things that we've gone in and added so that you can see really the detail with that, right? There are lots when we will see some of these micrographs and they color them purple, like a gram-positive, like grapes, because they say grape-like clusters, but like a gram reaction, and they are gram-positives. I kind of like that, right? I also like it right here with these guys being yellowish or golden colored, because aureus means golden. And here, with all of these golden little cells, I think it's kind of interesting. You can grow them on, it depends on the plates, all right? You can grow them on some types of media and like a nutrient auger and see the cultures, the colonies on these cultures or on these plates that have more of a yellow or kind of a golden tint to them, all right? If you had Staph aureus and Staph epidermidis on two plates, two nutrient auger plates, you should see a color difference between those colonies, all right? Epidermidis doesn't have that golden sort of appearance. So that's a little tidbit, a little fun fact about these guys, but I like it because it's kind of showing that right there, that they do have sort of that appearance. These guys, these colonies look very different when we see them on media compared to other bacterial cultures. This is tuberculosis, Mycobacterium tuberculosis. We have some mycobacteria that we work with in the teaching labs. It's not tuberculosis, but the appearance on culture media is pretty similar, all right? As far as these weird looking, shiny colonies, they're very irregular, right? You have gone through a lab this week and looked at some of this stuff and thought about some colony morphology, right? There are definitely some details as you look at this that maybe you're thinking about a little bit, right? We're not going to get bogged down with that in here, right? Not right now, but I bet you as you're looking at this, you're like, wait a minute, this is different, right? I'm wondering, did you see any sort of colonies that were like this kind of cauliflower-like is what I think as I look at it, except shiny. So it's cauliflower with butter, so it tastes good and you want to eat it, right? So tuberculosis is really kind of an oddball when we think about trying to grow these organisms in a lab, right? It is super hard to get it to grow, and it takes a long time to really get colonies on these plates, all right? Weeks for sure of incubation that it's going to take before you really get good stuff going on. If we're thinking real, patient situation, we could detect tuberculosis through a sputum sample as we acid fast stain it. Then we would want to do culture confirmation and do antibiotic testing after that. In order to cultivate it, this is what we think about. And there's a lot of different types of media that we're bombarded with in laboratories. We will start showing you some stuff that we talk about as like selective and differential media, things that help you weed out organisms or keep organisms. So you want some stuff to grow and you want to inhibit other stuff. There's all sorts of different types of media that we might throw out there to try to get organisms to grow. And if we really are thinking tuberculosis, Lowenstein-Jensen agar is a very specialized type of medium that basically is inhibitory to pretty much everything else. We really are focusing in on mycobacteria. So this type of organism wants to grow on these plates. There's a lot of, I'm assuming probably some antimicrobial agents. I should have looked up some details about this media. There's definitely things that are inhibitory to other bacteria because these plates or these tubes will be incubated again for prolonged periods of time. And keeping something like that, the potential for other things to maybe contaminate is an issue. You want to inhibit that stuff. So their growth on here, some of the shininess that we're seeing is the mycolic acid that they have in their cell wall. So on the outer bit of it, kind of a casing that helps to give them a leg up and lends to some pathogenicity as well. All right. That's also what makes the gram stain typically unable to penetrate in through those cells. Right. So Lowenstein-Jensen is a specialized media for growing tuberculosis. There are probably a few others that might tolerate this media and grow on it, but TB is really what we're thinking about. Yeah.
Do you happen to know like how the TB tests work? Where they
Sure
do it in your arm and you wait 24 hours? Like, how does that?
So a TB test, not this, right? This is a culture test, where we're growing it up, but a TB test where, and if you're thinking like healthcare stuff, many of you will be getting tested or have probably already got tested to see what's up, right? So TB test basically takes some antigen, it's purified antigen, so like a fraction of that. And we're going to come back to this later on when we start thinking some immunology stuff. But you could take a little bit of that, you inject it under the skin, and then it's more like it's a delayed reaction. It's not the same, but it's kind of like a poison ivy or poison oak, something like that. It takes time for those cells to recognize and stimulate a response. So if you have, it doesn't mean that you have tuberculosis, you could, but you've definitely produced a response against it, a cell-mediated response. And when you get that under the skin after 48 to 72 hours, I think is what they're looking for with that. If it's a big red welt, it indicates a positive reaction, and you've mounted a response, a cell-mediated response. That indicates exposure or potential infection. That's when, with the positive TB test, you would do more stuff like collect a sputum specimen, plate it out, see are they actively, you'd also probably do an x-ray. Probably do that first, and then try to cultivate it and see what's up. Good?
Yeah, thank you.
Yep. And we will definitely show some of that, some images of that. And I think, I don't think I know, if you military or if you are, international, if you've, from some other country, it's highly likely that you've been vaccinated against tuberculosis. And in those circumstances, you'd have a positive TB test. The vaccine will react with that. So you also need to know that as well, right? As far as what's happening. So, okay, here we've got an image, and we see all sorts of phylogenetic trees as we move through biology and think about stuff. I'm pretty sure we've seen this at a previous point as well, as we saw some of this diversity and talking about domains. We want to see some more stuff as we're moving down here, but we can see, and really the focus of this chapter are these two, the bacteria and the archaea, and a plethora of each of them. Numerous extremophile types of archaea, all sorts of diverse types of bacteria, and then the eukaryotes over here, we'll talk about them in the next chapter, right? There are a whole bunch on here we really want to think about as some potentials, right? And then really we'll get to ourselves later on as we start thinking disease and how these guys all impact us. It all really kind of fits together. So with this phylogenetic tree, we can see all sorts of names, genera out at the branches or the tips of these branches. And we can get some ideas as far as where we're at on this tree, as far as like some gauging of evolutionary time frame, if you will. All right. This one right here that we've put the little star on is one that is a very ancient type of microbe. This is Acetothermus, and it is something that's kind of, you know, like in the mix between these two, where it is something that is a high heat tolerant bacterium, not archaea, all right? And so also with this deeply branching, as it's clear down here toward the very bottom, as we see this last common ancestor, universal common ancestor, these guys are very ancient when we think about it, as opposed to up here at the top where we see like little animals over in eukarya. They're clear up here, very long ways away from this node down here that differentiates. All right, pretty cool. Dinococcus, these guys, I always just have to step back and wonder what in the world is happening here. Dinococcus radiodurans, they say the conand of bacterium. This is like it's like just crazy to think about this type of organism and how it could ever survive such inhospitable habitats. Dinococcus radiodurans is an extreme radiation lover. It's been found, it's been isolated in the cooling ponds in nuclear reactors where all of the fuel rods are maintained. Interesting, disturbing. How can it tolerate such high levels? I mean, thousands of times the amount of radiation that our body could withstand. These things, like I eat this for breakfast, right? Really pretty impressive, all right? And there are all sorts of ideas and research that's going into these guys and some other extremophiles, some archaea that might have some similar sorts of potentials. When we think about this, I mean, radiation, I would think is probably one of the top when we think extreme sorts of habitats, right? I just eat uranium, it's fine, right? That seems pretty extreme, all right? So there are all sorts of instances when we think about things like radiation resistance or the potential for communication and to tell one cell or another that there is a bad situation happening here, and these organisms have the potential with communication to let them know there are specific circumstances that maybe aren't so good for them. I mean, they obviously can tolerate, but how is it that they tolerate? And we're truly, you know, learning all sorts of things about these guys as we go. I don't have great details to spout off about them other than the fact that they are so resistant, impressive. Yes, it is. They do thrive in radiation. That question was probably 2 minutes old. I already probably got you there with that. All right. Yeah, so thriving in those types of habitats, really pretty impressive. Not the same, all right, but there was a professor here years back that was curious about this, all right, but not working with Dinococcus. She worked with the type of halobacterium. So another extremophile, but different, right? And the question was, are they resistant? Are there issues that they might have that could allow them to tolerate things like radiation? Physics has an accelerator, a linear accelerator that will have the capacity to bombard samples with high-powered electrons, right? Similar to thinking, can we tolerate, radiation is really kind of the idea with this. And they found all sorts of things like bystander effect, where you shoot this beam of high energy at a cell and it causes some serious damage. That cell starts telling other cells, this bystander effect. Holy cow, look what just happened to me. Prepare yourselves, right? Prepare to die, right? And they start mounting stronger responses and try to withstand these circumstances. Crazy, all right? Really pretty impressive. And she has since moved on to another university, but really pretty impressive stuff that they were working with.
Is this a type of cyanobacteria?
No, it looks green. It looks like it should be cyano. It's not cyanobacteria. It is it on my, oh my God, going the wrong way. Is it over here anywhere? I can't tell you where it's at. It's not cyano. I will do a little digging and let you know. Google it up. Where are you, Donna Caucus? Sulfobolus, this is a type of archaea. It oxidizes sulfur. It stores in these little granules sulfuric acid. I think that's awesome, right? So something maybe storing that up as a protective mechanism, maybe storing it up and utilizing that to degrade substances and pull them in as foodstuffs. Both potentials with that, I think, really pretty impressive. And just one other instance of really the extreme nature of some of those archaea organisms. This image, I don't think we're looking at the Great Salt Lake, but we sure could be, all right? As we see this, I did show you some images of the Salt Lake a little bit ago, maybe that was Monday. You can see here we've walled off a bunch of different areas, and as the water's evaporating, we're getting some salt within there. The colors that we're seeing are some of those halobacteria, and you can see some of them have more and potentially these greater concentration of salt that makes it more conducive to those guys growing versus some of these in the foreground that are more white, right? All sorts of potentials with this. Morton Salt Factory at the bottom of the Great Salt Lake does something very similar to this, walling off parts of the lake, letting it evaporate, and then going in and scooping it all up, right? Pretty interesting with that. This was, I think, non-majors biology 4 or 5 years back. It was a bit ago. I had a student that had all sorts of different fish. He had like saltwater fish tanks. He had all sorts of them, right? Crazy stuff. And he's growing, like corals and all sorts of stuff. And I teach some parasitology, and he's talking about all sorts of crazy aquatic parasites, and you get these corals and who knows what the heck's growing on them. All of a sudden fish are dying and whatever, and you've introduced some sort of crazy parasite because there's, who knows, an egg or something that's within all of this that you just transplanted into your aquarium. So he's talking to me about some of these things, and we start in lecture talking about some of this variation and halobacteria and pink sea salt and all of these things. And he recognized he's making up his saltwater stuff to put into his tanks. And then he starts seeing some of this pinkish stuff, the halobacteria, in not all of them, but some of the aquariums were getting some of this stuff in there, and really pretty interesting. And Now he's like, he's not a scientist, right? He's in non-majors biology. He wants nothing to do with it. And then all of a sudden he's like, wait a minute. I think I'm doing science, right? I'm like, well, I think you were doing science before you ever even got in here, right? That's good, right? So pretty interesting. And he's like, who knew I was growing these microbes? All right, I know that's the end of that hip exercise. We will maybe, I've got to stop sharing first. Okay. Any questions about that stuff? I'm going to switch over and introduce maybe the next chapter. We'll get a little bit into this. This has so many different things as we move through here. I mentioned a second ago parasitology, and it's something that I think is awesome and disturbing, maybe at the same time. All sorts of eukaryotes we could think about within that category of being parasites. Deep down, if we want to step back and think about a parasite, it's something that lives at the expense of something else. So it doesn't have to just be a worm in your intestines that we would say is a parasite. Any sort of pathogenic organism, any bacteria, anything like that that's robbing somebody is technically a parasite, all right? But when we really think about parasitology, we think about protists and we think about helminths, all right? The bacteria and the viruses that are also technically parasitic, we think about them differently and we just think of them as pathogens, all right? But in the big umbrella of what really is a parasite, there's all sorts of things, all right, that are parasitic, all right, from microscopic to gigantic, all right, and anything in between, all right. So a few of those we will think about as we move through and focus on some of these eukaryotes. There are definitely others that are not parasitic that we will mention in here as well. But we need to start with maybe one of the best, I guess it depends on who you're asking. Malaria is a parasite that has plagued humanity since the dawn of humanity, right? Truly, it's plagued people for forever, all right? And really pretty impressive to see some details about it. We're not going to go into great detail, but there is definitely some coevolution that's going on, and we see that with all sorts of pathogens when we interact with these things over the course of time. There's adaptations, right? And malaria adapts, and we adapt, and we try to combat one another, and we ultimately coexist, right? There are definitely some issues with that, right? But living as a part of us for forever has caused all sorts of impacts on humanity, right? And it's really pretty impressive. There are definitely circumstances where we see areas full of malaria and it has huge impacts. And then we look at other areas like the United States and we don't really think anything of it, all right? And that's maybe not so good, all right? Malaria was definitely a parasite that plagued the United States. It was the early, maybe mid 1900s, 1940s maybe, when we finally eradicated the mosquito that harbors this parasite from the US, right? So it's not, you know, it's something that's plagued us for a super long time, and it's only been gone from here for a short amount of time. I'm also a little disturbed because the mosquitoes that transmit it are back, we do see them. We don't see any of them right now harboring the parasites. But part of me thinks, maybe that's just a matter of time. And we'll be back to issues of malaria right here, all right? Definitely some concerns. We are going to talk more details about malaria when we get into the blood infections later on, and we'll talk some circumstances about really what's happening, all right? I technically feel like I could talk about malaria all day long, all right? But I'm not going to, all right? So these are eukaryotic parasites, protists, right? There's a handful of different types of protists, and how they get into us is definitely varied, right? Malaria is transmitted to us by mosquitoes. This is a sporozoite, which is a stage that's found in the mosquito, in its saliva. And when that mosquito feeds on us, when it takes a blood meal, it injects the saliva. It has all sorts of stuff in the saliva to keep the blood flowing. And it also potentially has some of these parasites. As it's injecting that stuff, it can inject the parasites into us. And then once they get into us, there's technically some steps that need to happen. And the parasite migrates into the liver, replicates out of those liver cells into the red cells. And that's what we're looking at right here. When we really think about malaria, we think about, for the most part, the cycle that's happening in the red blood cells, because that's really when we start seeing ill effects. Within your liver, you don't really recognize much at all right? But in the red cells, that's a different story, all right? And again, like I said, we'll talk more details with malaria later, but there are numerous species of malaria that infect all sorts of stuff, all right? There are a handful of species of malaria that infect us, all right? And with that said, there are variations with the type of species as far as how it impacts your red blood cells. Some of them target old red cells, some of them target young red cells, some of them don't give a crap and will target every aged red blood cell. That's the bad malaria, right? If it doesn't care how old your red blood cell is and it will just infect it, you got some bad news, right? So if they get in, all right, dependent upon the species, the parasite gets into the red cell, and it's kind of, interestingly, almost like a time-lapse photography as we move down there, and it's just a few cells that are all together, all right? When they first kind of penetrate in, that cell gets in, and these red cells, here's a bunch of normal red cells, non-nucleated, kind of dimpled in, it looks like. And then they get something in them. Here you can see little specks, right, where that parasite's just got in. Here it starts to form this ring stage. As it starts to grow, these parasites start feeding on the contents of the red cell. They start replicating. The tropozoite is the feeding stage, and it replicates and degrades the hemoglobin. You get all of these little granules, these speckles that we can see in here. And down below, as we are going through this process of replication, generating more parasites, it's this schizont stage where we're replicating and filling it up with new baby malaria parasites. That red cell will rupture, releasing all of these parasites to in turn infect new red cells, all right? And the details with some of your illness happen right at that point. As that cell releases the parasites, that's when our immune response is able to see them. Then they penetrate into the red cells, and then we don't recognize it. They hide, all right? And it sets the stage for the fever and chill cycle that results. Again, we'll think some more details about that stuff later. There are all sorts of potentials, all right? If we are traveling or living in these types of environments, that may set the stage to lending to a bit of protection, mosquito netting, netting or tarps that are impregnated with insecticides, clothing like long sleeves and pants, especially in dusk and dawn situations when mosquitoes are more active, will help to protect. Mosquito repellent, DEET-based insecticides, all harbor some levels of protection. There are also other things, medicines you can be taking if you know you're going into these environments that are going to be protective, right? So it's not like you're just walking in there blind, all right? We're not going to get bogged down with that right now either, all right? But there are definitely some differences with that. Okay, I will shift away from that, even though I want to talk more malaria, I won't. This next image is something that's, to me, a little bit unnerving. Ringworm is not a worm, even though it's got worm in its name. It's not. It was mistaken as such, and it kind of almost looks like maybe something like that on your skin. Or under your skin, right? It's also it's given the genus name Taenia, which means worm, but it is a misnomer. It's not a worm. Ringworm is a type of fungus, and this is something that's, I mean, we could call it a zoonotic disease. It's something that, you know, pets or animals can transmit to humans. It could be on their skin and then get onto you, or vice versa. It's something that is a bit of an issue. Fungal infections take time to treat. And it's something that if you don't fully adequately treat this stuff, you really don't get rid of it. It seems like it's went away, but it comes back. All right. It's like, you know, the misuse of antibiotics, and you take a week's worth when you're supposed to take 2 weeks worth, and it sets the stage for issues. But in this case, it's not a week's worth. You've got to take it for a month or maybe 2 months, right? It's a long duration of applying this stuff. And it kind of makes sense. After a few weeks, it looks better, right? I seem all fine and good. So you stop, but the fungus is still there, and it basically flares back up. And we'll go through some more details and talk about different types of Taenias, because it's more than just ringworm. All right, athlete's foot is a type of Taenia. Different circumstance, right? Slightly different, but similar as far as the fungi that may cause it, right? It just depends on where we're talking about within the body, right? Giardia, the next one up here. Oh man, it's one of my favorites. Giardia is a very common parasitic infection, right? In Idaho, for sure, it's the top of our list for #1 parasite, right? Probably in the whole United States, Giardia probably is the top of the list. And I would like to think, knowing that, we're in pretty good shape, because Giardia, in the big scheme of things, yeah, you're going to crap yourself, right? But not to death, right? I think you're going to be okay, all right? It's a little bit different, all right? If you've been infected with it, you're going to maybe think otherwise and say, that's bad news, right? But it's not as bad as it could be, right? So here's a whole bunch of these guys, and these are trophozoites. When we talk about these parasites, a trophozoite is just a general term that means the active feeding stage of these protists, right? So you'll see trophozoite all over the place. It's just when these guys are active, right? They also will, not all of them, but lots of these protists will form cysts. And the cysts are typically what is infective. You ingest the cyst, And the cyst in turn hatches out as the trophozoite and starts to feed, right? It depends, right? The malaria that we started with, it's the sporozoite that's infectious as that mosquito feeds upon us. And once it gets into us and it starts infecting, it's technically a trophozoite as well when it's feeding and replicating in those red blood cells, right? So these guys, here we've got a whole bunch of them in the intestines. Here's just an image of one of them. And we're looking at the dorsal side of these guys, of this one right here. Gigantic sucker on the bottom of them, all right? You can't see it here, right? So they attach to these cells with this big sucker, all right? This is an electron micrograph, but if we stained it up, you could see two nuclei within here. The suction cup, as you're looking at all of it, it kind of almost looks like a little smiley face, right? And then a bunch of little hairs poking down off of them. You're not smiling though if you've got it, all right? So these guys, and this is in a gerbil, but it could be in your own gut. Giardia gets into us through contamination. So like drinking water that's got some of these cysts in there. Hiking puts you at risk if you decide you want to drink some water right out of the stream, all right? Lots of people are schooled on the fact that you should not do that. You shouldn't just drink this water, even if it looks like it's fresh, right? It looks crystal clear, right? You should still filter it or boil it, somehow sterilize it, because Giardia should be on your radar. And if it's not that, there's definitely other things like some bacteria in the water that it could also make you sick, right? But Giardia is an issue all over the place, right? And in turn, however it happens, and we'll talk more Giardia later, it doesn't have to just be consumption of water, but usually it's coming in contact with water that has those cysts in there, and somehow, one way or another, you ingest those cysts, right? Usually with these types of infections, you've ingested a few cysts. Those cysts get in, they hatch out, they start feeding, they start replicating, and then all of the sudden, out over the course of a few weeks to a month, their population has exploded. Now you've got massive amounts of these parasites in your intestine. We call these guys duodenalis or intestinalis, along with lamblia. But the duodenum is one of the main places where these guys will take up residence. And once they've truly replicated, they can cover virtually every single cell, all right? And now your potential for nutrient absorption is dramatically minimized. You've got serious diarrhea, you got some serious ill effects, all right? But it's maybe a month after you drank that water from the stream. You're not thinking about that trip, right? That's not what's on the radar. You probably think Taco Bell, right? Something like that gave me these issues, right? But it wasn't, right? So I'm going to quit right here, but I will tell you these trophozoites, if you've got like serious diarrhea going on, that's what you're going to be presenting with. That's not what would be infective. It needs to be the cysts, and that doesn't present with diarrhea, right? There are patients that have serious issues with this, and in a single diuretic stool, they can release upwards of 14 billion of these parasites. That is impressive, and that's what I want to leave you with. Have a great weekend, guys.